So I'm going to be talking about this thing called the Topological Morphology Descriptor, or as I like to call it, the TMD for short. And what this thing does is it takes the data of a neuron and then turns that neuron into its collection of branches in the hope that that'll be a little simpler to analyze. So when I'm talking about neurons, what I'm really talking about is measurements of neurons. And the way that I usually think about neurons is that someone's gone into a lab and they've measured a bunch of points along the neuron, and then they also know where those points are connected together with edges. And moreover, they know where the nucleus of the neuron is. And once I have that, I can think about a neuron as this sort of data structure, where each point has some sort of notion of distance from the nucleus of my neuron. And once I have this, I can rip this apart in a principled way until I have just a collection of branches. And the way that I do this is I pick a branching point, okay, and then I look at the two branches above my point, okay, and I pick one branch that goes the furthest up. Now both of these branches go to 3, so I can pick either branch, okay, and so then I get something like this, where I've ripped off the branch to the right because I chose to pick the branch on the right. And now there's still one branching point right here. And this time when I look at the two branches going up, one of them goes all the way up to 3, whereas the other one only goes up to 2. And the rule in this sort of case is I always rip off the shorter branches. So I'll always rip off 2 to 1 in this situation, leaving me with this collection of intervals, this collection of branches in my neuron. And now that I'm here, this is a much simpler data structure, and hopefully I can do some analysis with this, and I can understand the distribution of the sorts of branches I have in my neuron. So in summary, I've gone from this really complicated data structure, or kind of data, the shape of a neuron, into something that's a lot simpler, just the neuron's branches, a collection of intervals. So maybe I'm in a situation where I have a collection of intervals representing one data set, and then I have another collection of intervals representing another data set, and I want to say how similar they are on a computer. How might I do that? Well, one way of doing it is using what are called persistence images. So what you do is you start with your collection of intervals, and then you imagine each interval as a point in the 2D plane. And the way that you do this is you think of the point as having x value of its bottom endpoint and y value of its top endpoint. So for example, this interval from 1 to 2 gets sent to 1, 2 in the plane. And then once I have these points, I'm going to turn these points into a function by the following procedure. First, what I will do is I'll imagine a Gaussian hovering ominously above each point that I've just made. Okay? And I'm going to weight these Gaussian functions by how far above these points are from this diagonal line y equals x. And after I've done that, I'll add these Gaussian functions together, and that'll give me some new function f. And then what I can do is I can look at the values of my function f over a grid in the plane, some finite grid, okay? And then I get a grid of numbers. And now, if I have some other collection of intervals, I can then get the grid of numbers for that collection, and I can compare the numbers from each collection. And that's how you compare a collection of intervals, or one way of doing it. And this grid over here is what's called a persistence image. And this is really just one idea from the field of topological data analysis, which is really all about taking complicated geometric data and finding some sort of way to turn it into something that we can analyze on a computer while still interpreting it back to the shape that we started with.